All right. How's everyone feeling? Yeah, I know it's definitely stressful, but hopefully you'll start to feel better as you get through it and as you get closer. But one step at a time, one one subject at a time, one idea concept at a time. You guys got this. All right, I'm gonna get started. My slides, yeah, definitely. I was just about to give my intro, Jonathan. Um, I was planning on sharing my slides with Josh to upload to the portal after my lecture. Would that work? Would that work? And if in the future you want me to share my slides in advance, um, I can do that too. Share them now. Okay, I have questions in my slides. Okay, I have questions in my slides with the answers, so I don't want to reveal the answers. Um, if everyone's okay with waiting like 2 minutes, I can quickly like delete those and try to share those slides with you. All
Just email
right.
them afterwards.
Sorry?
I said just email them afterwards.
Okay, it looks like there are quite a few people who want the slides now to annotate them. So I'm just gonna quickly give me like just a couple minutes and I'm gonna drop those, take care of that and drop that in the chat. So sorry. Yeah, thank you everyone for your patience. Just 2 minutes. All right, thank you everyone so much for waiting patiently. All right, let me know if anyone has trouble opening those slides. All right, let's get started. So let me just start by introducing myself. I'm Prachi. My pronouns are she, her, hers. I graduated from UC Davis just this past June. So June 2021, I was a global disease biology major. A little bit about my MCAT journey, in case it helps to it helps to understand what other people's journeys might look like. I actually took MCAT twice. I studied for it like 3 times. And yeah, the most recent time that I took it and I'm done, I took it in September 2020. And I'm also applying to med schools this cycle. And a little bit more, I also took the MCAT with accommodations. And for anyone out there who is either taking it with accommodations or wants to or is planning on taking it with accommodations, I know how daunting of a process that can be. So I'm happy to talk about that with anyone during office hours or after class. And just a little bit more about me. I'm from the Bay, from San Francisco Bay Area, and outside of like application stuff and things like that, I'm very involved in a lot of like advocacy and anti-oppression organizing work and research specifically related to curricula reforms and curricula development in health and biological sciences. Yeah, so that was a little bit of a tangent, but I know sometimes it helps to hear a little bit about the person you're hearing from for 2 hours. And just on a side note, if you hear like any noises or barking in the background, I apologize in advance for my dog. All right. So just an outline of what today is going to look like. I sort of split today up into two broad sections. So one is the first part talking about broader genetic concepts like Mendelian genetics, non-Mendelian inheritance, recombination, we'll get into that. And then the second part, talking more about DNA, replication, nucleic acids, more of the fun nitty gritty. And just a few disclaimers. As you can probably already tell, this lecture is going to be really content heavy and there's a lot of information to get through. So we can get everyone on the same page here. And so I ask that you bear with me, and for our lecture that's in 2 weeks, which is the genetic expression lecture, I'm going to get into some more example questions, and hopefully we'll have more time for passages then as well. And you can also come to my office hours next week on Wednesday. I'm going to send Josh the link so he can put it up there, but I'm planning on having my office hours Wednesday at 12 p.m. and I'd be happy to review any passages with you or any practice problems. And I wanted to emphasize that having this really solid foundation of core biology content is going to be really, really important for doing well in biochem. And you can't get away without knowing, especially this core content. And yeah, while I'm talking, feel free to drop a message in the chat if you have a question, and I'll try to monitor everything of like 5 screens open right now. But if I miss your question, feel free to jump in and unmute. All right, let's get started. Percentile, a lot of us. Okay, I don't feel comfortable sharing my percentile, but I will say that I scored well enough to be able to teach this content. And yeah, I understand. I definitely understand your nervousness, but I'm hoping that you can put your faith in me, and we can learn together. All right, so let's get into, okay. Sorry, a lot of chat messages coming in. I want to make sure I'm reading them all while keeping everything going. All right, so Mendelian genetics. I'm gonna run through some brief vocab here. But so what's a gene? A gene is a unit of hereditary information. And you often hear about alleles too. So an allele is 1 or 2 more alternatives of a given gene. So we have this beautiful diagram on the side over here. And with our two, let me see, is my annotation working? Oh, sorry about that. Okay, just wanted to check that I'm able to write on here. Yeah, so you can see that our alleles in this Punnett square and this example here are capital R and lowercase r. So each of those are alleles representing two different variations of the gene that codes for color in this flower. And the locus of a gene is the position on an allele. So you can see my cursor. Okay, I was not drawing, I didn't have my cursor, I wasn't using my cursor, but I will annotate as I go. So a locus is the position on an allele in the chromosome. So like if we have our chromosome here, the locus would be the location where you would see the allele on the chromosome. Okay, yeah, no worries. I'm gonna try to, I like gesture a lot with my hands, but I'll try to make sure I'm like writing on the page as well as I go. All right, so actually, before I go to the next slide, can anyone tell me what the genotype and phenotype are of these flowers? Feel free to unmute. It's the flowers over here. Purple and white, awesome. Yeah, exactly. So, oh my gosh, I'm sorry. I'm having a little bit of trouble with the screen share here. I'm not sure why. I think it just crashed on me. Sorry, everyone. I'm so sorry. Technical difficulties happen. All right, if I have issues with that again, I will follow your advice and yeah, stop screen share or stop my camera. Okay. So yes, our genotype and our genotype is the genetic makeup of the organism. So that is the actual like RR and the capital R, lowercase r, these things that we see here. That's our genotype. And then the phenotype is the outward appearance of what the flower looks like. So it being purple or white, that's the phenotype. Okay. And both alleles, so what is it? So we have these different combinations, right? What do these different combinations mean? So when we have a homozygous organism, that means that we have both of the alleles are the same. So they're both either dominant or they're both recessive. And then heterozygous means that you have two alleles for a given gene, that two different alleles for a given gene. So you're going to have one that's dominant, you're going to have one that's recessive. And there's also hemizygous, actually. So that usually occurs with the X chromosome in males. It's when there's only one allele present for a given gene. So that means that if there's a male with the X and Y chromosome, and there's a gene on the X chromosome and it's not present on the Y, that means they're hemizygous. There's an allele that's on the X chromosome that it doesn't have a counterpart to. So now if we take a closer look at this diagram, you'll see that each generation has this specific label. Like this one says paternal generation, this one says F1, this one says F2. So across most of the Mendelian genetics questions, you're going to see these same labels be used. The paternal generation refers to organisms that are homozygous for the dominant and recessive genes. So we have our purple and white flowers with capital R, lowercase r. And then the flowers in the F1 generation, they're a cross between that parental generation. So you have all of your flowers are going to be heterozygous. They're all going to have that capital R, lowercase r genotype and a purple phenotype, because in this case, purple is the dominant the dominant allele. And then in the F2 generation, that's a cross between the F1 generation, so a cross between those heterozygous organisms. So you have, because of that, you're going to have a variety of different resulting options. So you'll have your two homozygous organisms, homozygous dominant, homozygous recessive, and then you have the heterozygous ones. Someone asked, would this be like X-linked genes in terms of hemizygous? Exactly, that's exactly what I mean. So X-linked genes are also known as like hemizygous genes. All right. Hemizygous? Yes, it's on the previous slide. Let me just go back. Yeah. All right, thanks for everyone helping out in the chat. Okay, so Mendel came up with these basically laws in like after doing all of his like experiments, he came up with these laws. It's pretty much similar to what I've already gone over in the last slide. But these are like the official laws, and you'll hear them mentioned, so I wanted to go over them. So there's the law of segregation, which means that the alleles of a gene are separated and passed on to offspring in the next generation individually, retaining their identity. So they're not, the gametes resulting from meiosis are only carrying one allele for any inherited trait. And then there's the law of independent assortment. Okay, there's the law for independent assortment, which means that the alleles of a gene will separate into gametes independently of alleles from another gene. And the inheritance, so that means that the inheritance of one gene does not impact the inheritance of another gene. So for example, like if you have gene R and gene W, they won't impact each other. Okay, someone asked for me to draw out the laws. That's, okay, let me see what I can do. So law 1 is saying that the gametes resulting from meiosis only carry one allele for any inherited trait. So let's say we're like drawing out meiosis. So we have R. This is like before it's split. So now we're going to split it into, this is our diploid. So now each of our gametes resulting from meiosis are going to have only one of the alleles. So it's not going to have both capital R and lowercase r. It's only going to have one of them. And then the law of independent assortment, that one's, so let's say you have like gene. I apologize for my bad handwriting. I don't know what's going on today. It's usually better than this. But let's say you have gene W and gene R. So they are going to, according to this law, which we'll talk more about this later too, about like what exactly happens in real life, or like what are the different possibilities. But for Mendelian genetics, this is the law that we follow, that if you have gene W and gene R, they're not going to, it's no more likely for okay, let me give a more concrete example. So let's say we have someone who's heterozygous for gene W and heterozygous for gene R. This is one person. And when they're again, they're undergoing meiosis. So it's equally likely to have a gamete that's WR, like capital W, capital R, and it's equally as likely to have someone who's homozygote, like the gamete. This is the gamete. Sorry. Let me draw these out. So I don't know if this is making sense. So what I'm saying is that these, it's equally likely to have all of these different gametes with the different alleles all separated out. So it's not more likely to have for you to have a gamete that has like the dominant gene for W and the dominant gene for R. Is this making sense? Sorry, I would draw out like a better diagram. Not really? Okay. Let's do this. Okay. So again, we have gene W. Actually, let's do something other than W because it's hard to tell the capital or lowercase. Let's use D. Gene D and gene R. So let's say you have an individual. This is like the individual's genotype. They have, let's just make this easier. Let's say they're homozygous dominant for gene D and recessive for gene R. Sorry, heterozygous for gene R. Similar, eye color is a little bit different because that's caused by multiple alleles. But yeah, okay, let me talk about more like in terms of Mendel's things. So like Mendel had like his P's and some of them were wrinkled, and some of them were yellow. So we have a gene for it being wrinkled or not, and then we have a gene for the color. So the gametes that result from that. So basically what I'm saying is that it being wrinkled or being yellow, they don't have anything to do with each other. They're not going to impact each other. It's not like if I have a pea that's yellow, it's more likely to be wrinkled. It's just going to separate, they're going to separate equally. So we can essentially split this up into two different things. Even though we're looking at two genes, we look at them as separate genes. Does that make more sense? Okay. Let me know if it doesn't make sense, and I can try and find another way to explain it. Okay. Yeah, in the future, I will add like a slide about independent assortment because I know that's a tricky one to sort of comprehend without a visual. All right, so that's most of what I have for the content of Mendelian genetics. Yeah, so wrinkles doesn't influence color, exactly. Yeah, someone asked, so wrinkles don't influence color, yeah. Okay, so if everyone's okay to do a practice problem, I'm gonna give everyone 2 minutes for this question. And okay, sounds good. All right, if you have an answer now, feel free to drop it in the chat. And if you're still working, just don't look at the chat for a little bit. Okay, do folks still need more time? All right, no worries. Let's try walking through this together. So I heard someone typed in D, someone said B. Does someone want to tell me how they approach this question?
I guess I could maybe explain how I approached the first part of the question, because it says red is dominant to brown. So I did 2 big R's and then 2 little R's for brown. 2 red beetles are crossed, which means, and then there's 31 red and 9 brown. So that would mean that, so you didn't do it that way. I think I did like double R, or 2 big R's and then heterozygous.
Okay.
Because in my head, it's like a 3-to-1 ratio of red to brown, basically. So that would mean like there's some heterozygous red and then 1 homozygous brown. But I could be wrong.
Okay. So this question is kind of sneaky. Because yeah, there's so much going on in this question. It really is, it takes a little bit. So okay. Sorry, so many messages coming in. Okay, okay. A lot of folks are on the right track here. So yeah, you're looking at your F1 generation, and it's asking about the probability of having, because, okay, probability that both red and brown beetles will appear in F2. So it's asking for you're only looking at the red F1 beetles. So that's something that you need to think about. What is the probability of having a red F1 beetle? And then from there, so let me write this out. It's asking about so I'm trying to find the best way to explain this without giving away the answer completely. Okay. So you have your Punnett square, right? And we know that you're on the right track, Marissa, that we know that it's probably, since you're generating both red and brown offspring, you probably have a heterozygous Punnett square. So what do you get from that? This is what you get. You get 3 red and you get 1 brown. And if you sort of look at this ratio, it pretty much follows that 75-25. So that's the first part of it, right? Understanding that this Punnett square. But then it's asking if 2 red F1 beetles are crossed. So then at that point, you're only looking at the red ones. You forget about the brown completely. If 2 red F1 beetles are crossed, what's the probability that both red and brown beetles will appear in the F2 generation? So what do you need to get both red and brown beetles in the F2 generation? What genotypes do you need? Exactly. This is the genotype you want. You want to have, this is the only cross that's going to get you both of those. So then you look at what's the probability of getting a heterozygous beetle from only the red beetles. So you have these three options basically, and you're thinking about the probability of getting first one heterozygous beetle. So that, yeah, some folks said it in the chat. Oh, yeah, some folks said it in the chat, and that's 2 out of 3. And then you do the same thing for the other one, and that's also 2 out of 3. Because remember, you don't want just one heterozygous beetle, you want 2. So to get 2, you need to think about the probability of both of them being heterozygous. So then you have your 2 over 3, 2 over 3, and then you multiply. And then that gets you the answer, which is 4 over 9. How's everyone feeling? Okay, someone wants me to explain it again. I can do that. Is there anything in particular that didn't make sense? 2 over 3. Okay. Okay, let me go to the next slide because I actually have it drawn out a little bit more nicely. So I got the 2 over 3 because I looked at the number of red beetles in this cross. There are 3 red beetles. And what I want to get this answer, to get both red and brown beetles from the F2 generation, I need the F1, I need the beetles that we picked out to be heterozygous. Because let's say I did like capital R, like I did homozygous dominant with a heterozygous. All of the possible, they're all going to be either the offspring are all going to be homozygous dominant or heterozygous, which means that we're not going to have any brown beetles. So the only way for us to get both red and brown beetles is for us to pick out the heterozygous beetles from these 3. So that's how we got the 2 over 3, because 2 out of the 3 red beetles in the F1 generation are going to be heterozygous. Okay, then how did I get the 4 over 9? So to have offspring, you need 2 beetles. So you need 2 beetles that are heterozygous. You need this cross of heterozygous. You need this heterozygous cross. So 2 over 3 is the probability of us getting 1 heterozygous beetle. We need 2 heterozygous beetles to get that F2 generation of red and brown beetles. So that's why we multiply 2 over 3, because in probability, right, when you're looking at the probability of two things happening, you're going to multiply. Does that make more sense? Yeah, you're right. Someone said that the probability of heterozygous is 1/2. That's true. But this question is sneaky. This is a sneaky question. They said that if 2 red F1 beetles are crossed, so that they're telling you that you're only thinking about, you know that these beetles are red, you don't know what their genotypes are. Okay, how would this be different if the note wasn't there, that assume Mendelian inheritance. You wouldn't really be able to, if you did not assume that this is Mendelian inheritance. There might be like unknown variables and factors about the way that, and I'm actually going to go over that soon, about what non-Mendelian inheritance looks like. Okay, someone said, assuming that we already got 2 reds, the probability that each of those 2 reds is 2/3, and they have to get 2 of them, so they have 2/3 chance in the first 2. Yeah, exactly. Yeah. Yeah, thank you. Okay. Everyone feeling better about this? Let me know if you still have any questions. Yeah, it's sneaky. Yeah, exactly. You feel better once you learn to catch their little tricks. But yeah, this is actually a question from the AMC section bank. And I didn't get a chance to find a question that's not a spoiler from a full length, but on these topics. But other questions that you might want to be on the lookout for, that, or just like if you're thinking about what does this look like in an exam, you might get questions about the probability of a certain outcome, ratios of like all outcomes. So like, for example, you might get a question, if we're looking at this F1 cross, you might get a question about like, what's the ratio of red to brown? That one's fairly easy, 3-to-1. And then you might get questions about the probability of an outcome for a hybrid cross. I didn't want to overcomplicate this during our lecture, so I didn't put an example of the dihybrid cross, but if you have questions about that, we can talk about that during office hours. And then other things just related to Mendelian genetics, like you want to be careful about what's an inheritable mutation and what's not. So you might have like a question that asks you about what something looks like, what the offspring of someone might look like if they have this mutation. But you need to think about, is that mutation, is that genetic, or is that something like, for example, like, is it cancerous that was not like a genetic mutation? It's something that you've developed in a somatic sense. Okay, let me catch up on some of these chat questions. Someone asked, do you know any good genetics Anki decks that you can share? Honestly, I don't really think that Anki is the best way to go for genetics, at least Mendelian genetics, unless you're like trying to get your vocab down. I don't know if Anki would be the best way. I think really just practice problems and getting familiar with how to manipulate these numbers and like understand what different things mean and what different generations mean. That's probably the most important thing. But if anyone else has any suggestions, feel free to drop those. Yeah, I've definitely had other people also think that this question meant it was a dihybrid cross, but dihybrid, usually you're thinking about two different genes. So the reason that we don't count the RR is because it's homozygous. Yeah, exactly. Sorry, I lost who said that. Yeah, Jonathan, exactly, to answer your previous question. Okay, yeah, no worries. Someone asked me a question about the difference between hybrid, and dihybrid and viability. If I want to keep going and get through all this, at least most of this content, yeah, you can come to office hours and ask me, and I can't give you an answer off the top of my head, but I can look into it. And if you email me as well, I can try to get you back with an answer. I'll put my email in the chat at the end. All right. I'm gonna keep going. But yeah, feel free to keep asking questions as we go. All right, I'm switching gears now to non-Mendelian genetics. So far we've been talking about like the idea of complete dominance, which is the idea that like a dominant allele completely overrides a recessive allele. So if you have a dominant allele, the recessive allele and its phenotype do not matter. But there are a few other forms of inheritance that Mendel didn't really talk about or like experiment with as much. So there's the idea of incomplete dominance. And that's the idea that a dominant allele is expressed at a reduced ratio. So what does that mean? Also, by the way, incomplete dominance is also known as partial dominance. You might see it come up in that way sometimes. So muted color. Okay. So I want us to think about, this is actually something I always forget. And then when I was creating this lecture, I realized again, and it hit me. So I think this is a really interesting concept to get into a little bit more. So, okay, it's muted color. So we have, in this example, we have this red homozygous dominant flower combined with a white homozygous recessive flower. And then with our heterozygous, instead of getting a red flower, we're getting a pink flower. But I want us to think about a little bit more about why does that happen? Like, what's happening there? Like, what are the mechanisms potentially behind that? So what's happening is that it's less, or like you have less red. It's muted because there's less of the dominant genetic material present. Exactly, Sophie. So if we were to like fill out this table we have here, our genotype for red is capital R, genotype for pink, capital R, lowercase r, genotype for white, lowercase r. And then if you look at the amount of gene product, how much red allele content is there? Let's say our one allele is X. So over here we have 2X. Over here we have X. Over here we have 0. So basically the lack of a second dominant allele in the pink flower. Oh, that arrow is going the wrong way. The lack of a second dominant allele in the red flower, in the pink flower, means that there's insufficient genetic material to produce a red flower. Does that make sense? Everyone feeling good about that? I'm just checking in as we're going, just because I want to make sure that my explanations are making sense to everyone. Okay. So I'm going to move to the next non-Mendelian inheritance concept, which is codominance. So codominance is when there's independence of allele function. Neither allele is dominant or even partially dominant over a phenotype. So this is just an example of if you have like a heterozygous genotype. It's not actually presenting as if like the red is dominant over the blue. They're both exhibiting themselves in equal parts. So basically in codominance, both alleles are simultaneously expressed in heterozygous individuals. So the most common example of codominance, the most classic example, is blood type. So the alleles indicate, so in the case of blood type, the alleles indicate the antigens that the red blood cells are presenting. So these little guys over here are antigens. And so you'll notice that for the genotype that's A and B, it also has a phenotype of type AB instead of just type A or just type B. All right. Yeah, and then there's also the option of not having any. So codominance is interesting in that way. Okay, and lastly, there's, I talked about this a little bit briefly earlier, but lastly, there is sex-linked inheritance. So that's often when there's a sex-linked gene, or when there's a gene that's located on a sex chromosome, so the X or Y chromosome. And alleles can be dominant or recessive, or can have like any form of inheritance too. There can be like there can be codominant and incomplete dominance in sex-linked genes as well. So sex-linked genes are more often located in the X chromosome, since the X chromosome is larger and holds more DNA material as well. You'll very, very rarely see a Y-linked, a Y chromosome sex-linked gene. And then on the opposite side, like the normal genes that we usually talk about are autosomal genes. Those are genes that are not located on any sex chromosomes, either the X or the Y chromosome. Okay, next I have what seems to be everyone's favorite practice questions. I have a couple of practice questions here. So this is a pedigree. I'm going to give everyone a few minutes to answer it, a couple minutes, but feel free to message me if you have any questions about how to read this or anything else. Okay, sure, yeah, let me just explain how to read this. So with pedigrees, the blacked out, so actually, let me start by explaining the circles and the squares. So the circles are usually represent females and the squares represent males. And then the blacked out, so female, male. And then the ones that are like blacked out, like these ones, those are the ones that have the syndrome. Yeah, and in some pedigrees, you'll see like it might look like this, like they'll have half of it colored, and that means that it's like heterozygous. But not for all. Usually this means like heterozygous dominant, like this is the dominant allele. Yeah, box sickles male, yeah, exactly. And circle is female. All right, I'm going to be quiet now so folks can read the question. Sorry, so someone asked which one is dominant and which one is recessive. You don't know for this question, you don't know. Okay, it sounds like a few folks have their answers. Does anyone want to explain why they picked a certain answer? I want to hear everyone's reasonings. Yeah, exactly. That's actually the right answer, and that's exactly the reasoning. So good job. Yeah. So yeah, it's sex-linked dominant. Sex-linked dominant is the inheritance that cannot explain the pattern. So basically, if so you want to think about this in terms of the X and a Y chromosome, especially if this is sex-linked. So female XX, male XY. And for any, if you think about the scenario where you have a male who has the syndrome and a female who does not have the syndrome, or who could be who could be a carrier for it, for all of their female offspring, this X chromosome is going to be the one that each female offspring inherits. They're also going to inherit either one of these, but they will definitely inherit they'll definitely inherit the father's X chromosome. So if this were to be dominant, then we would see that every female offspring would have this pattern, would have this syndrome. Yeah. Let's see. I saw some folks, someone said A, someone said C. Does that make sense to everyone? Anyone need me to explain like why it's not a specific answer? Okay, so let's talk about A first. If this were sex-linked recessive, yeah, it's always like the small things. Someone said that they didn't pick up on the cannot in the question. Yeah, I feel it. I always mess up questions for the tiny things like this. Okay, let me explain A real quick, and then I'll catch up on the rest of the chat messages. So for A, if this were to be sex-linked recessive, we would see, which it could be, because that's actually what it's asking, but we're saying that why is it or is it not sex-linked recessive? So let's start by looking at the females again. So you have XX. So this female could have inherited this one from the father, like definitely inherited one of the X chromosomes from the father, and inherited either this X chromosome or this X chromosome from the mother. So one of those. So let's say, for example, that the mother is a carrier. So let's just say C. This is, or actually, since it's syndrome Z, let's say Z. Let's say that she's a carrier, and the father also has an X chromosome with the Z allele, and this daughter inherits the Z X chromosome from the father, but inherits the normal X chromosome from the mother. And if it's sex-linked recessive, it explains it. That makes sense. That she doesn't, since this is the recessive syndrome, since it's recessive, she doesn't have the syndrome. Makes sense. And then same for the female that does have. My Z's are looking like 2s. Same for the female that does have it. She inherited the syndrome, the X chromosome with the syndrome from both of her parents. So, okay, it makes sense for the females. Doesn't make sense for the males. You would have the male inherit their Y chromosome from the father, and then one of the X chromosomes from the mother. Yeah, so we have a male without the syndrome, and we have a male with the syndrome because they inherited one of these. Does that make sense? Yeah, someone asked if B was true, then females would have the syndrome because they would be getting the dominant they would be getting the dominant X chromosome from their father in generation 1. Yeah, exactly. All right. Are we good to move on? I have another question for us. Okay, I'm going to go to the next one. Oh, no, that's, oh yeah. Oh wait, no, that's the answer. Pretend you didn't see that. All right, this is unfortunately my last question, but hopefully you'll like it. All right, I'm seeing some A's and some C's. Let's walk through this. Does someone want to tell me, let's say we say that the allele for colorblind and for being colorblind is XC and the other one is normal. So what would the genotype be for the heterozygous mother? Yeah, exactly. So we'll have one of the X chromosomes would be carrying the colorblind allele. And then what about the father? The father is not colorblind. Exactly. Sophie, correct. Exactly. All right. And then what about Dylan? Dylan is colorblind. All right. Thank you. Thank you. Thank you. All right. So is it possible then to have, what would the, let's say if there's a female who's colorblind, what would the, Masa, sorry, I hope I'm pronouncing your name correctly. Someone asked if it's XC for the mother. So the mother is heterozygous. So that's why you would have, so one of the X chromosomes would have the colorblind allele, but not the other. Okay, yeah. And so for a female being colorblind, it would be XC. 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 Exactly. So from what we have, looking at the mother's genotype and the father's genotype, is it possible to get an XC? Or like, what is the possibility? I kind of revealed it right there, but what's the possibility of getting an XC genotype for the female offspring? Yeah, Michaela said impossible. Yeah. Yeah, and the mom can give the XC or the X. Yeah, exactly. So that makes it 0, impossible. That's if you want to look at my very hastily put together pedigree. All right, is everyone good on that? Anyone have any questions? Also, if I miss your question, please feel free to shout it out. Someone asked if I have any genetics passages we can do together. So I think for today, I'm not even going to be able to cover the content that I had planned. So if you want to do genetics passages together, I'd be totally down for it. But I think office hours would be the best bet for it because yeah, already I'm probably not going to get through most of the content. All right, awesome. All right, switching gears again. No, so some of these are AMC questions. The first one was an AMC question. The last two that we did, those were not AMC questions. All right. I don't remember where I got them from, in case you're wondering. Okay, so recombination. What is recombination? So, actually, backing up, before I get into recombination, let's do a little bit of chromosome recap. So after DNA replication, our cell is going to have 4N. That's like the amount, the amount of DNA material it has, and basically before replication, we're going to have 2 chromosomes. This is, by the way, this is like someone who's this is not like meiosis or anything. This is I'm switching over to like thinking more about like replication. But yeah, so each person has a set of homologous chromosomes, which means they're the same chromosomes or chromosomes that code for the same genes, but they might have different alleles, and one is going to be from the mother, one's going to be from the father. After replication, you're going to have each of those chromosomes replicate. So you end up with sister chromatids, each of which have the same genetic material. These two have the same, and this one has the same. And again, together, those are called homologous chromosomes. And sorry, just making sure I'm checking the chat. The full set of homologous chromosomes with each chromatid having a sister chromatid, basically this whole replicated thing, this is all called a tetrad. Okay, so I know we just talked about Mendelian genetics a little bit ago, but contrary to Mendelian genetics, specifically contrary to the law of independent assortment, genes that are located on the same chromosome might not display independent assortment. So you can see in this first picture, we have this top one we have over here. We have gene A on one chromosome and gene B on another chromosome. They're able to independently assort. It doesn't make a difference. They don't impact each other. That still follows Mendelian genetics. But if we have gene A and gene B on the same chromosome, you're more likely to get, this is what I was getting at earlier. You're more likely to get A and B being together. Like you're more likely to have this combination, these two being separate, than you are to have like, oh, Yeah, you're more likely to have that than you are to have like capital A and lowercase b and capital B and lowercase a. Yeah, someone asked if a tetrad is both chromosomes. Yeah, the tetrad is both homologous chromosomes. Let me go back one slide. Yeah, so it's once it's been replicated. So it's the homologous chromosomes that have sister chromatids. I'm sure there's a better, a nicer way of explaining that, but yeah. All right, so does that make sense? Basically, we still have independent assortment with genes that appear on different chromosomes, but if they appear on the same chromosome, they're more likely to be linked. They're going to be together. You're less likely to have this combination, but it's still possible. It's not totally impossible, because you might remember in meiosis, in prophase 1, you have crossover. There's this crossover event that makes it possible to have that different combination of alleles on the gene. So going back again, recombination, that crossover event, the crossover event makes it possible to have that capital B lowercase a, but it's less likely than it would be to have like the capital A capital B. Okay. So again, this is saying pretty much what I just said, but talking about a little bit more about distance. So the farther apart two genes are on the same chromosome, the greater the chance of a recombination event between them. So like for these two, there's such little distance, it's very unlikely for there to be a crossover right here. So that's why you're going to see this these original chromosomes stay together most of the time, and this crossover event happening less frequently. But if you have a little bit more distance between them, so over here we have more distance between the two genes A and B, it's you're more likely to have a recombination event between them, and end up with these recombinant gametes. So just to clarify again, these parental gametes are referring to these chromosomes, or like these two genes staying together and there being no recombination. And then recombination meaning that there's like a crossover event here. So you end up with these two together and these two together. Okay. Someone said they were a bit confused with the 99%. That was just like, that was just sort of an arbitrary number. Oh, okay. So this one, if you compare this slide 22, slide 23, these are much closer together. These are much farther apart. So if you think about it, like the more distance there is, you can have a recombination, a crossover event here, crossover here, crossover here, crossover here, crossover here. There's more possibility for having a crossover between them if they're further apart. If they're closer together, you have two things that are right next to each other, it's less likely that there's a crossover right between them. The 99%, it's an arbitrary number, like saying that most of the time you're not going to have them when they're really close together. It's much less likely to have that crossover event, which means that they're going to be like linked, more closely linked. So these are the parental genes. So this is the same. This is the same as this. So it's more likely to stay the same and not cross over because they're so close together. Okay. I'm going to walk us through an example, and hopefully this will make a little bit more sense. The 99% was just sort of a random number to give us an idea about what kind of an impact distance makes. So we often look with recombination, you might see questions about the degree of linkage between two genes. So like in the previous one with A and B, how closely linked are they? And you can tell that with understanding like how much distance is there between the genes, but how do you actually do that experimentally? So that's what I'm walking through here. So if we look at, so these are the two parental, these are our two parental phies. So just for reference, PR means that it refers to the eyes. So PR plus means you have the wild type red eyes. PR means you have the purple eyes. And then VG refers to the wings. So VG plus is the wild type normal wing. VG, and then VG without the plus is the vestigial wings, so the smaller wings. So if we're doing this test cross, can someone tell me what this might be a little hard to do over chat or even verbally saying it, but we can try this out. If folks can tell me what are the possible recombinations. Let's say, so we're doing an F1 test cross. So we're doing F1 to get our second generation. So what are the possible recombinations or just possible combinations of the gametes that you can get? So gametes means you're going to have one chromosome because these are the two homologous chromosomes. So we're going to have 4 different gametes. If folks can tell me, what are the alleles going to be on each of those? Yeah, so 4 different gametes. What are the alleles going to be on those 4 gametes? Each line, by the way, represents like a chromosome. Okay, so someone said PR. Oh, it's not letting me write in that corner. It's strange. Okay. So we have PRBG. Oh, PR plus. Okay. I got it. Okay. PRBG. Yeah, and Sophie, you got the 4 different alleles that we're looking at. Oh no. Okay. And then we have the recombinant ones. So this is a recomb. I should have left more space on this slide. This is the parental. PR plus, BG plus, this is parental. And then we have PR, BG plus recom. Yeah, I gave it away by going one slide ahead. Okay, I hope that makes sense for everyone. Yeah, so we have our 4 different gametes, and these first 2 came directly from the parents. And these second 2 are the recombinant events. So you have the PR plus and VG. That's what you have over here. And then you have the VG plus and the PR. That's what you have over here. Yeah, so this does actually violate independent assortment, and that goes back to the idea I talked about a few slides ago, right here, where they're on the same because because the alleles, sorry, the genes PR and VG exist on the same chromosome, they don't independently assort. Okay. Does that make sense? Does everyone understand how we got to these 4 gametes? Okay, good. So now we're going to do our test cross, and then we get these numbers. We did our test cross, and we find out that most of our offspring are parental. They have the same they have the parental chromosomes that we taught, the parental gametes that and parental chromosomes. But there are a few that have this these recombinant chromosomes, the gametes that we talked about earlier. So yeah, the test gives us the different results for the different offspring, and basically we can then use these numbers that we got to come up with a metric to help us identify the linkage between the PR and the VG genes. So again, like how far are they? How closely linked are they? How likely are they to independently, or how unlikely are they to independently assort? So then we come to this metric, the recombination frequency, which is basically the distance between linked genes. And you get that by putting the number of recombinants over the total number of offspring and then multiplying by 100. So if we do that for what we have over here, this is what we get. We add the 2 recombinants, we add the numbers of all the offspring that have the recombinant genes, the recombinant chromosomes. And then we put it over the total number of offspring to get this number, and then multiply it by 100 to get the map units. And that's the metric that they use to say that, to like identify the distance between 2 genes on an allele. So they're not actually looking at the chromosome and saying that this is how far apart these genes are. So that's like, this is sort of how they would experiment to find that out. So on an exam, you might get a question with, maybe they'll give you these numbers already. They'll ask you to come up with a recombination frequency, or they'll, maybe they won't ask you to give a number, but they might ask if, let's say, for example, this is 0. What would that mean? Or let's say, actually not 0. Let's say all of these were evenly split. And you don't know if the 2 genes are on the same, you don't know if the 2 genes are on the same chromosome. That would probably tell you that they're not on the same chromosome, because they're able to, they're basically independently assorting at that point. So yeah, map units indicate like how far or close something is. So a larger number means that it's going to be further apart. Yeah, the decimal moved. Someone asked why the decimal moved. Decimal moved because what? Decimal moved because we multiplied by 100.
I have a quick question.
Mhm.
For the last question that I asked, is there like a set amount of map units that we should look out for? Or
No.
is it arbitrary?
Yeah, it's there's no like, you won't see too, like, this is biochem, so you'll definitely see numbers, but you won't necessarily have too much of this like calculation heavy thing. Think about like, I guess it comes down to like understanding the fundamental of it. Do you understand what more map units means compared to less map units? That type of thing.
So they'll basically give us like maybe like 2 examples, and then we'll have, or like 2 different map unit numbers, and then we'll be like, okay, these are closer together and these are further apart.
It might not give you the map unit numbers. They might give you like the data, but yeah, you can like sort of compare between 2 different things.
Got it. Thanks.
Yeah. And for that, you might not even have to like go through the calculation. It comes back to like that fundamental understanding, right? Like if you have, let's say you have 2 sets of experiments, right? They're doing like the same thing, but they're looking at different genes. And let's say one of them, you get a lot more recombinants. That likely means that it's the 2 genes that you're looking at compared to the other genes are even further apart. Exactly. Oh, wow. Oh my God, it's almost 2:30. I am not even halfway through my lecture. Okay, we'll figure it out if we can't get through this. I think questions are always really important. We'll find another time to get through everything, because I don't know if I'll be able to fit everything into my genetic expression lecture. We'll see. All right, so moving to population genetics. So there are a lot of different concepts for population genetics, but the most important one that you'll see most commonly on the MCAT is the Hardy-Weinberg law. Sorry, laptop's dying. So the Hardy-Weinberg law states that the frequencies of alleles in a gene pool will not change over time, provided that a number of assumptions are true. For example, that there's no mutation in the gene pool, there's no migration in and out of the population, there's no natural selection at work, there's random mating between individuals, and that the population is sufficiently large to prevent random drift in allele frequency. So those are basically the conditions under which the Hardy-Weinberg equation works. All right, so I'm just going to go over what the equation looks like and what the different components of it mean. But if folks want a practice passage on it, I would recommend passage 5 in the genetic information chapter. And again, happy to go over that in office hours if folks want to go through it together. Oh, okay. Thank you, Iman, for that response. I did not know that. Okay, so for the Hardy-Weinberg equation, yeah. So the equation states that it's similar to like our binomial equations in that you learn in like algebra. So there's actually a version of this that's not written, but let me write that out so I don't forget about it. So the PQ, so let me just read this out loud. P squared plus 2PQ plus Q squared equals 1. And then P plus Q also equals 1. So in this case, we're saying that P, if we have alleles, A, capital A, lowercase a, dominant recessive, P represents the proportion of the capital A allele. The Q represents the proportion of the recessive lowercase a allele. And then naturally, it would make sense that if you were looking for homozygous dominant, which is AA, you would square that. You would multiply the probability of the proportion of the capital A allele twice. And that gets you the proportion of the homozygous dominant individuals or organisms in the population. And then same for the heterozygous recessive, and same for the heterozygous and same for the homozygous recessive. So you have, so since the Q represents the proportion of lowercase a or recessive allele, 2PQ and Q squared, so 2PQ is going to represent the proportion of heterozygous individuals, Q squared is going to represent the proportion of homozygous recessive individuals. And then there's this other version of this equation, which is P plus Q squared equals 1. So same thing here. So basically, if you factor this out, you'll get the same thing. But what does that sort of tell you? It tells you that if you have just P, like with these three different versions of this equation, if you have just P, let's say like P equals 0.2, you can figure out what Q is. And from there, you can figure out what P squared is, what 2PQ is, what Q squared is. So you just need one of the numbers. Yeah, exactly. So then Q would equal 0.8. And then Q squared, if we were to do this out, would be 0.64. PQ would be 0.04. And then so on and so forth. But it makes it really simple. But yeah, so you might just get like one metric, and you might be asked to find like, so if you're given like the proportion of A, capital A allele in the population, you have to find like the proportion of heterozygous individuals in the population. It's completely doable, but you just need to get comfortable with manipulating the equation. Yeah. So even though it sounds perfect, it sounds really neat and pretty, but again, it only works under these like specific conditions. And in real life, allele frequencies do change over time, and mutation, migration, natural selection, non-random mating, and random drift all do occur. So that's, I guess, one of those small things to keep an eye out for if you get a question that doesn't, that might either say that you don't have mutation, or like, sorry, you do have mutation in the population. You know, Hardy-Weinberg won't work for that. It won't work for that population, and same for any of the other factors. But in my experience, that hasn't really been something that you need to like memorize hardcore. It's been pretty apparent when Hardy-Weinberg needs to be used. But again, that's my experience. MCAT always throws those questions at you. Okay, let's see. There's 30 minutes. I was thinking of taking a break, but seeing as how short on time we are, I'm gonna see if we can power through and get through as much of this content. So I apologize for not being able to give everyone a break. All right, so switching gears again, a little bit more, getting into DNA. So I just wanted to touch on the central dogma to provide us with the necessary context for this next section. But I'll be talking more about like translation and transcription in the genetic expression lecture in 2 weeks. But for now, we're just looking at DNA replication, which is indicated by this curved arrow that we have, the DNA polymerase. But yeah, before I get into how DNA replication works, we need to better understand the components that make up DNA. And I guess also before that, some other things to highlight. Just reminder: DNA through transcription. Oh, sorry, translation. We get RNA, and then through transcription, we get protein. But yeah, we'll be getting into those details in a couple of weeks. Did I get it flipped? Oh, I always do that. That's on me. Transcription, translation. Okay. Sorry about that. Yeah. Okay. Are we good to keep going? Oh, that's a good trick, Sophie. I was going to come up with a trick for the genetic expression lecture to remember that, but I think maybe I'll just steal yours then. Okay, so nucleic acids. There are 5 nucleic acids that make up the genetic code, DNA and RNA. for both DNA and RNA. And I recommend being able to identify and distinguish each of these structures and knowing like what groups they belong to. So we have adenine and guanine, which are the purines. And then we have cytosine, thymine, and uracil, which are the pyrimidines. And so thymine and uracil, so thymine exists for DNA and then it gets switched out for RNA, where it's uracil. So in terms of hydrogen bonding, where you have your two strands of DNA and the nucleic bases on each side are going to hydrogen bond to each other, A bonds to T, and it also for RNA, it bonds to U, and then G bonds to C. And that's another thing is that G and C, the bond between G and C has 3 hydrogen bonds, while the bond between A and T and A and U have 2 hydrogen bonds. I'll talk about that in a little bit too. So yeah, like I said, A and G are purines, while C, T, and U are pyrimidines. And so a way to remember that, I think some folks might be dropping mnemonics and ways to remember in the chat, but the way that I remember it, at least, is that cytosine has a Y and thymine has a Y, and so does a pyrimidine. So cytosine, thymine, pyrimidine. And then A and T then, A and G then are purines. And then thinking more structurally, purines have 2 rings, have these 2 ring structures, and pyrimidines have 1 ring. So a way to remember that between the purines and pyrimidines is that you can imagine like a pyramid over that ring structure. So that you can, I can't really draw it out, but you can imagine like a pyramid existing over the ring structure and it being like one pyramid. But that doesn't exactly work for purine. So pyrimidine pyramid. Yeah. Ag, pure silver. Pure. Yeah, that's a good one too. And then you also want to think about like being able to distinguish between the different nucleic bases. So you might be asked to like recognize like what nucleic acid is this, or you might have to sort of apply that knowledge. So guanine has a if you're comparing, so I'm going to compare the purines together and then the pyrimidines together, just because the 2 rings through 1 ring is pretty straightforward once you have that down. So for guanine and adenine, guanine has this it has this carbonyl group, whereas adenine does not have any carbonyl groups that are coming off of that ring structure. They both have this amino group, but that's the similarity. And then here, actually, I'll keep that circled. And then cytosine has, between the pyrimidines, cytosine has a carbonyl group and an amino group, whereas uracil has 2 carbonyl groups. And thymine is special. Thymine is the only one with a methyl group. So I don't have any mnemonics for that off the top of my head, or that I was able to come up with. But if anyone thinks of one, feel free to share. Yeah, also let me know if you want me to repeat that. And these red H's down here, these ones, they all represent where the base of the nucleic acid bonds with the rest of the nucleotide structure. So it's really important to know also which hydrogens in the nucleic base hydrogen bond with each other. Does that H have a specific name? So technically that H doesn't exist because that's where your ribose bonds. I'll talk about that in like 2 slides. but no, that H doesn't actually really exist. In reality, I think it should be an R if you're looking at it in the structure of the nucleic acid. Yeah. Unless, obviously, I could be wrong. There could be a name that I just don't know about. But nothing that you need to know that, yeah. So yeah, it's important to know which hydrogens in the nucleic base hydrogen bond with each other. So again, A and T, adenine and thymine, have 2 hydrogen bonds, and guanine and cytosine have 3 hydrogen bonds between them. So you can sort of see, yeah, this diagram points out the locations where there are like which either nitrogens, oxygens, or hydrogens are going to be involved in that in hydrogen bonding. So if you want something for Anki, this could be something that you can like screenshot this and like remove the arrows, or you can just have an adenine and try to figure out which are the hydrogens that are involved. So if you want to sort of think about it and like figure out which ones, which hydrogens or nitrogens and oxygens are involved, that's a good way. So one thing that I noticed, at least, is that the hydrogen in all of the amino groups, so we have our amino groups here. Actually, there's no amino group in the thymine, but the adenine, cytosine, guanine, the amino groups in all of them, the hydrogen is involved in hydrogen bonding. And then the oxygen in each carboxyl group, sorry, carbonyl group, not carboxyl. The oxygen is also involved in hydrogen bonding. And then, especially for guanine and cytosine, the nitrogen that's in between those, that's sort of sandwiched by the amino group and the carbonyl group, is also, sorry, the hydrogen, that's hydrogen or the nitrogen that's sandwiched between the two, is involved in hydrogen bonding. That's not like the cleanest way to explain it, but I think it's just one of those things that once you practice it and you see it a few times, you'll be able to more easily identify which ones are involved. And I think it also helps like sort of trying to take the nucleic acid and sort of like superimpose it onto like this diagram and figure out in that way which hydrogens or nitrogens or oxygens are involved in bonding. Yeah, and again, it's like the nitrogen that's inside of the ring that's involved. Okay, so talking more about the structure of the nucleic acid. So yeah, before this, I was talking about more of like the bases and how the bases interact with each other, but there's more to the nucleic acid than just the base. So we have our ribose, which is our 5-ring structure. It's also called sometimes sugar or a pentose sugar. And what we see here is actually this is actually deoxy. Wow, I did not plan out that spacing. This is actually deoxyribose because it doesn't have an oxygen here. So the way it is right now, this is what you would see for DNA. But if we were to add, if this were to be an oxygen, that were an OH instead, if it were a hydroxy group, then this would be RNA. So the ribose is different between the DNA and the RNA. And for the OH, it's actually a little bit more unstable than just the hydrogen. So it's a little more reactive as well. Yeah. And then we have our phosphate group here, and I will talk more about that in our next slide. So what does DNA replication actually look like? What happens at the nucleotide level? So you're going to be adding nucleotides from the 5' to the 3' direction. So I'll talk more about this if I end up getting to DNA replication. But basically, you're always going to synthesize, you're always going to write from 5' to 3'. And you're always, again, I'll talk more about this later, but I wanted to mention it. You'll be reading from 3' to 5'. And the reasoning, if you are actually not the reasoning, but if you want a way to remember that, the way that I always remember it is that writing when you learn to write. Here, let me write them out. When you learn to write and when you learn to read, you usually learn to read before you learn to write. So you learn to read when you're 3. So that's 3' to 5'. You learn to write when you're 5. Not necessarily, but you learn to write when you're older. So 5' to 3'. That's the way that I remember it. It works pretty well for me. So how does the synthesis actually work? So this is our dNTP. So it's actually not our it's our nucleotide with an addition of 2 phosphates. So deoxynucleoside triphosphate. So what happens is that this is like our DNA sequence over here. The oxygen on the ribose, or deoxyribose technically, the oxygen on the deoxyribose over here attacks the first phosphorus in our triphosphate, which kicks off these 2 phosphate groups. Okay, let me know if I get anything wrong. It happens. Okay. So that's how you synthesize, that's how you generate this bond over here. And you've basically kicked off 2p. And this is actually an energy releasing reaction because there's a lot of tension, but when you have three phosphate groups, there's a lot of tension. So when you break that bond, you actually release some energy. What are the name of the phosphates? I don't remember. If I tell you off the top of my head, it's probably going to be wrong. So I don't remember the names. I think it helps to know. I think it helps to know like how to name the different like phosphates and things in a chain. Yeah. Let me make a note of that. Okay. Yeah, feel free to email me if you don't find the answer, and I can get back to you with that too. All right, let's see. Let's do time check. 3:45. Okay, I think I can probably get through most of replication. We can do this. Okay. So DNA replication, the actual mechanism. So eukaryotes have, yeah, so I wanted this to be like, eukaryotes actually have multiple origins of replication, and prokaryotes generally just have one origin of replication. And so we need to remember that in this diagram, one of the things I want to emphasize is that DNA strands are anti-parallel, which means that they're built in opposite directions. That's why during replication and translation, you read a strand and write a strand in an opposite direction. So that gets back to the whole like writing or synthesizing from 5' to 3', and then reading from 3' to 5'. And hopefully that will make more sense as we go on. So I think one of the important things to think to like if you're looking for things to memorize related to DNA replication, I'd say those are the enzymes, the enzyme functions. And overall for the idea of DNA replication, just like understanding how it works, what direction things happen. And I think those are the more important things. So helicase is our enzyme here. It unwinds the DNA, and the topoisomerase is going to reveal supercoiling ahead of the replication fork. So if you think of like a rubber band, like if you're twisting it, it gets or like if you have a twisted rubber band and you're like untwisting it from one direction, it's going to keep getting more and more twisted at the other end. So that creates a lot of tension. So the topoisomerase sort of reveals and loosens that, and it decreases that tension that's created by the untwisting. And then one other thing is that replication can be bidirectional. So we can have replication going in both directions at the same time. And these single-stranded binding proteins over here are actually a prokaryote things. They're found in E. coli and can be bound to, and are usually bound to single strands of DNA just to stabilize them. Someone asked about helicases. So helicase is the actual thing that's working on unwinding the DNA. Okay, so what happens next? Next, primase actually initiates the DNA replication process by laying down these RNA primers that are about 5 nucleotides in length. So not very long, just like a short sequence just to help get things started. And that recruits DNA polymerase 3. And DNA polymerase 3 comes to this RNA primer and then starts reading the DNA from the 3' to 5' direction. So let's say we have like a DNA polymerase over here. It's going this direction, and this is like our current DNA sequence. We have 3' over here, 5' over here. So it's reading the sequence 3' to 5'. And then it starts synthesizing new DNA strands from 5' to 3'. That's the actual strand that it's synthesizing. So it does that like based off right after or onto the primers, the RNA primers. And then this process, because you have all these primers that you put down, this creates lagging strands, and it also creates Okazaki fragments. And the leading strand that we have over here, it can be continuously extended in the 3' direction. So your helicase keeps going, and the leading strand, it can just keep extending this replicated DNA. And then this lagging strand, it starts and then it hits a primer. Things have already been replicated, so there's no need for it to keep going. And then for Okazaki fragments, because it's going in like the opposite direction, it's because you're going to be synthesizing from 5' to 3'. It keeps moving, your helicase keeps moving, and you need to keep putting down primers from the 3' to 5', 5' to 3' locations. So you end up with a lot of different fragments. And then what happens to those fragments and what happens to those primers? DNA polymerase 1 then comes in and removes those RNA primers and adds on DNA nucleotides in its place. So it has this exonuclease activity. So it takes these out and it replaces it with DNA, because remember, we have the difference between the RNA primer, the RNA and the DNA. There's two things. There's the nucleic base, which is uracil versus thymine. You're gonna have uracil for RNA, thymine for DNA, and then you also have the ribose, which has a hydroxy group an extra hydroxy group in the RNA, and just has a hydrogen for the DNA. Yeah, Okazaki fragments are basically, so lagging strand generally refers to, it generally refers to, it's a little confusing because sometimes you hear this being talked about like in the bidirectional, in like bidirectionally and sometimes unidirectionally. But basically, yeah, those fragments that you have, those are going to be those are the Okazaki fragments. And then you still have, so going back to your DNA polymerase, once your DNA polymerase has replaced these RNA primers with DNA primers, there's still like gaps that are left between it, because you still haven't, you've replaced this with like a blue your DNA, but you haven't linked these two nucleotide, or these two bases. Okay, let's check. Okay, so I wanted to do a recap of the enzymes with everyone. So if folks want to shout out or message in the chat, if they can remember the function of each of the enzymes, I can write it out, and then we can just like check ourselves that way. Okay, helicase unwinds, awesome. Primase adds DNA primers, or sorry, RNA primers, yes. Okay, what about DNA polymerase 3? Okay, that's one of the things it does. What does it do after reading the strand in the 3' to 5'? Yeah, exactly. Okay, what about DNA polymerase 1? So RNA polymerase 1 removes the RNA primer and it adds DNA nucleotides in its place. I should just say replaces. Yeah, so the thing about DNA polymerase 1 is that it can only add like short sequences of DNA nucleotides. So that's why polymerase 1 and polymerase 3 are not interchangeable, because DNA polymerase 3 can go on for many, many base pairs. But that's not the case for DNA polymerase 1. Okay, what about DNA ligase? Yeah. Someone said seals Okazaki fragments, joins nucleotides. Yeah, exactly. Perfect. Why have I lost control? There we go. Okay, so that's the summary. If anyone wants to, if that helps anyone. Let's see, we have 4 minutes. I have one small thing that I can quickly go over, and I think we might finish just on time. And I can stick around for like 10 minutes to answer questions. Okay, so I wanted to quickly mention some of the theories of DNA replication. So I was going to put this before, but I wasn't sure we'd have time for this. So initially, when they were like figuring out how does DNA replication work, there are a few ideas over what is the end product of replication. So there's the idea of conservative replication, that basically the after replication, the two DNAs that you have, one is the original and the other is a complete is completely different. It's completely replicated. Both strands have been replicated. And then semi-conservative says that we have our original, and one of the strands from the original is in each, like, is in that each output. So you have like our blue one, so there's one blue one in each. And then you have one new fragment in each as well. And then the dispersive theory says that you're going to break that the two outputs that you have, each strand is a mix of new and original DNA. But as we just went over, in the end, it's actually, you're going to have semi-conservative. So in the end, after replication, the two DNAs that you have, one is going to have, both of them are going to have one original, one new, one original, one new. And I would recommend looking into this experiment because I've seen it come up a few times on full length and on the exam. And I've heard that it's a pretty common one as well. But it's like the experiment to tell if, like, what is the way that DNA replication happens. And I think it's in the book as well. So what they did is that they used nitrogen isotopes, so N14 and N15, to mark the DNA. They used nitrogen isotopes within the DNA. So if you were to do, if this conservative method theory of DNA replication was correct, you would end up with, and like you go through this whole process, you let them replicate, and the new thing that, like, let's say this is the N14, and then the added base pair, the added nucleotides that they put into the solution are N15. If the conservative theory was correct, you would see two different strands. Once it's like centrifuged, you'd see two different weights. Like one would be N14, one would be N15. And then what we actually see, because what happens in real life is semi-conservative, both strands are both DNA sequences have like one N15 strand, one N14 strand, one N15, one N14. So you just have one band in the centrifuge. And then for the dispersive one, where each strand is made up of new and old nucleotides, you would see like multiple layers over here, many layers, because they would basically each DNA sequence would be a different weight. Yeah. So I've had multiple questions come up on that, so that's why I wanted to put that in there. Someone asked if semi-conservative would be 14.5. Yeah, that's correct. You're right. Or, well, not. Sorry, I need water. 1 second. Also, if you need to head out right now, feel free to. That's pretty much most of the content. I made sure to, yeah, I can go back and explain some stuff over again too. But so if you look at the weight of it, like if you look at the mass of it, it would be like average, so it'd be 14.5. But in reality, like each nitrogen would still be one isotope or the other, because it's together. Like if you were to say like, what is the percentage of like N14 isotopes and what is the percentage of N15 isotopes, it would be 50-50 for semi-conservative. Someone asked me to, I'll go back and explain Okazaki fragments since we're done with the slide, but someone asked if I could explain dispersive on the slide again. So, wow, I'm really losing my voice. I was not expecting this. Is it multiple bands of N14.5? Not quite. So let's see. Let's say you have this dispersive band. I'm going to just make the N15 areas thicker for our purposes. So let's say you have two dispersive. Yeah. So the idea with dispersive is that you have some of the original and you have some of the new. So the new one would be N15 because you added N15 to the solution. So you have N15, and then the next nucleotide is like N14 because it's the original. Next nucleotide is N15. That is something like that. So let's say you were to let's say this is evenly split. Let's say we just keep going and it's like N14, N15. This is like 50%. So this is 50-50. And if we were to say like, what is the average mass of all isotopes in this one strand? It would be like 14.5. Not saying there's an actual isotope that's 14.5, but we're averaging the numbers. And then for this one, let's say, because it's like very random, there's no, it's not like it's, sorry, actually, let me change that a little bit. Let me change that. Let's say it's a little off. Let's say this is 14.4. This is 14.4. And let's say its complementary strand is exactly the same. It's exactly the same. But you have another one that was replicated alongside this. I hope this is making sense. That one would be 14.6. You won't actually get a question like asking about these numbers, but I'm trying to give you an example here to make this have this make more sense. But because of that, you don't have, when you put this in the centrifuge, you're going to have one DNA like sequence or one strand whole completed replicated DNA that's 14.4, and that's going to show up a little bit higher. Then you have one that's 14.6 that's going to show up a little bit lower. And for like all of your different, because it's not just going to be like one copy of a replicated DNA, you're gonna have multiple. So you'll have multiple and they'll all be different. You might have like one that's 14.7, 14.8, one that's 15, one that's, or probably not 15 actually if you're going with the dispersive model, but so basically you'll have a large variety, which is why you'll see a lot of different bands. So in reality, it might just look like one thick band. Yeah, exactly. So they asked, it's not necessarily 50%, whereas semi-conservative has to be 50%. Yeah, exactly. But it's also like dispersive it's not just like 50%, it's thinking about the two different strands in your DNA. It's almost like crossover in some ways, but not, I shouldn't use that word. I don't want to confuse anyone. But semi-conservative, one strand is completely new, one strand is completely original. Dispersive, it's a mixed match. Like one strand can have both new and original. And yeah, it does. And because of that, it doesn't have to be 50%. Okay, I'm gonna go back to Okazaki fragments. I have to admit, Okazaki fragments are not my favorite thing. And they're not my strong suit. So I will do my best, but if I can't answer your question, please give me your email and I will, I'll try, I'll send you an email and get back to you. Someone asked about which enzyme does the nucleotide repairs and corrections. I don't remember that off the top of my head. I was thinking of including it in this lecture, but I cut it for time. I can email you and get you that information, but sorry, I don't remember off the top of my head. If folks are interested in like nucleotide repair and corrections, I think it might be ligase, but I just want to be sure before getting back to you. Yeah. But if you're interested in nucleotide repair and corrections, I was planning on just skipping that completely, but I can see if I can fit it into my genetic expression lecture. Okay, someone asked me to go over Okazaki fragments again. Right. I hope they are still here and I didn't make everyone wait too long. So, yeah, so your helicase, let's think, I think this might be easier if I should have included a unidirectional example in here, but let's think about this unidirectionally. So our helicase is going this direction. This is 5 prime, 3 prime, 3 prime, 5 prime. So our primase can only place. So I'm hoping, Jonathan, to answer your question, I'm hoping that I can fit the rest of the information into the genetic expression lecture. If it turns out that I cannot, I will definitely talk to Josh, and if there's interest in another lecture on covering more material, thank you friends for looking that up. But yeah, okay, more lectures. Yeah, if there's I think I covered most of the important material here too. So a lot of this, I think if there's anything that I missed, I think it's doable to study it on your own. But yeah, happy to help folks out. Okay, so coming back to this, let me change the color of my pen. So primase can add the strategies on, someone asked about strategies on how to approach biopassages. I do have a review session that I have from earlier that I can probably talk about, where I go through a few tough passages. I can't say necessarily that it's strategies, it's just like exposure to a variety of tough passages. But yeah, I was thinking about doing that. Okay, sorry. So I'm going to focus on Okazaki fragments. All right, so our primase adds it adds nucleotides 5 prime to 3 prime. So it's going to add it this way because it's you're looking at the replicated strand at the replicated strand. So the replicated strand, this is 5 prime. Three prime. Draw one here. 3 prime. Sorry, this is not the neatest diagram. Okay, so then your DNA polymerase is going to, DNA polymerase 3 is going to, we're gonna jump on this primer and start generating, reading 3 prime to 5, 3 prime to 5 prime, and generating 5 prime to 3 prime. So it'll go this direction. So this is going the same direction as the helicase is going. But over here on this side, you have 5 prime, 3 prime. You're going to be generating in the opposite direction because you're generating from 5 prime to 3 prime and reading. These numbers are getting messy, but I'm hoping this is making sense. So you're generating then 5 prime to 3 prime, but now this is in the opposite direction of the direction that replication is going. So replication is going this way. Replication is going this way, but now you're generating DNA going, you're generating the replicated DNA going the opposite direction. So at some point then you need to put down another primer. You need to go back and put down another primer. As this opens up more, you need to put down another primer and then start generating this direction again. So that's how Okazaki fragments are generated, because you're not able to generate in the direction that replication is going. So you have to keep adding more, RNA primases, RNA primers, and generating in the opposite direction. So you end up with a lot of broken fragments as you keep adding primers and you keep generating opposite to the direction that the overall replication is going. Does that make sense? Yeah. Yeah, you still end up with like lagging strands on your parallel strand, because yeah, you're going to obviously have a few RNA primers, but you're going to have this like repeated thing only in the anti-parallel strand and on the strand that's going like 3' to 5', or yeah, 5' to 3', depending on which direction you look at it. Yeah, glad I could help. Yeah, lagging strand gaps, Okazaki fragments, the gaps between all of them, they're all filled by ligases. Yeah, glad I could help. Anyone else have any questions related to any of the content today, or anything that you want me to cover in the future, anything related to that?
Can I ask something really quickly? I actually have 2 questions.
Yeah.
One was like the specific role of RNA primers. Like, is that just the so it like knows where exactly it needs to go to start replicating?
Yeah.
Okay. And then the other was, I don't know if you're going to go over this another time, but something I was always kind of confused on is antisense and sense strands. If you could go over that quickly.
Yeah, so that's like actually this, if I remember correctly, I think that's basically the same concept as anti-parallel and parallel strands. So I should have left a blank slide on here. Let's see, what slide has the most space? Okay. Yeah, so basically your 5' to 3' strand is the sense strand. And the 3' to 5' is going to be the antisense. Yeah, and you hear it called by different terminology. Like someone had talked about anti-parallel and parallel. So that terminology is also used. Yeah, so basically that's where, like, when we think about replication, it's the antisense strand where the Okazaki fragments are created.
Okay.
And yeah.
And then the anti-parallel would also be antisense, right? Just to make sure.
Yeah.
Okay, perfect. Thank you.
Yeah, and so antisense and sense is usually, I've heard it talked about more when we talk about transcription and translation, because that's when you think about like you're only transcribing one of them, you're only transcribing, I think you're only transcribing the sense strand, not the antisense strand. And that's where we talk about like, okay, does the RNA that's created, that's like, that matches up with, oh, sorry, I got it mixed up. You're transcribing the antisense strands, your mRNA that's created matches up with the, it looks like very similar to your sense strand. Yeah. 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 DNA replication happens during interphase. Yep. All right. I actually have to head out. But yeah, if anyone has any other like last questions, I can answer those. But yeah, thank you everyone for sticking around, and thank you for being, yeah, of course. Thank you for being such active participants, everyone. Yeah, I have office hours next Wednesday. Yeah, I'll let Josh know so he can let everyone else know. Okay, someone asked about